Hi, everyone. Imagine boiling a bacterial cell for hours, exposing it to radiation, drying it out completely, or leaving it dormant for years, and then watching it come back to life when conditions improve. Most living cells cannot survive those kinds of extremes, but some bacteria can. Certain bacteria have evolved one of the most effective survival strategies in all of microbiology, and in biology for that matter, the endospore. Endospores allow cells to essentially enter a highly protected dormant state that can survive environmental conditions that would kill most other organisms. In this lecture, we'll explore what endospores are, how they form, and why they are so important in both nature and medicine. By the end of this lecture, you should be able to explain what endospores are and describe their role in microbial survival, and describe the processes of sporulation and germination. An endospore is a specialized survival structure produced by certain types of bacteria. Unlike a normal active bacterial cell, an endospore is metabolically inactive, meaning it is not actively growing, dividing, or carrying out most normal cellular processes. As you look at the diagram on this slide, notice that the endospore has multiple protective layers surrounding the core of the cell. These layers help protect the DNA and other important cellular components from environmental stress. The endospore core contains the bacterial genome, along with ribosomes and specialized protective molecules. Around that core are thick protective coatings, including the cortex, spore coats, and outer layers that provide remarkable resistance. Endospores are extremely resistant to heat, drying, radiation, chemicals, and other harsh environmental conditions. It is also important to understand that endospores are not reproductive structures. One bacterial cell forms one endospore, and when conditions improve, that endospore can germinate back into one single active bacterial cell. So the purpose of the endospore is survival, not reproduction. Endospore formation, called sporulation, is usually triggered by environmental stress. When conditions become unfavorable, such as nutrient limitation, drying, heat stress, or other harsh conditions, certain bacteria begin the process of converting an active cell into a highly resistant endospore. As you follow the diagram on this slide, the process begins with asymmetric cell division. Instead of splitting into two equal daughter cells, the bacterium forms a smaller compartment called the forespore inside the larger mother cell. The bacterial chromosome is copied, and one copy becomes enclosed within the developing forespore. Next, the mother cell engulfs the forespore, surrounding it with additional protective membranes and layers. As sporulation continues, thick protective coatings called the cortex and spore coats developed around the core. At the same time, the developing endospore becomes highly dehydrated. Water content decreases dramatically. Metabolism slows to extremely low levels, and the cell enters a dormant state. Eventually, the mother cell breaks apart, releasing the mature free endospore into the environment. If favorable conditions later return, the endospore can germinate and return to an active vegetative cell. As sporulation progresses, the developing endospore becomes one of the most resistant biological structures known. At the top of the slide, you can see the major stages of endospore development, including the formation of protective membranes, the cortex, and the protein spore coat. These layers act like that biological armor around the cell. The transmission electron micrograph on the lower left shows just how thick and complex these protective layers can be. Notice the dense outer regions surrounding the darker internal core of the endospore. One major factor that contributes to resistance is dehydration of the core. The internal environment of the endospore contains very little water, which helps stabilize proteins and DNA and reduces damage from heat and chemicals. Endospores are highly resistant to boiling temperatures, drying, radiation, disinfectants, and many harsh environmental conditions that would rapidly kill active bacterial cells. On the lower right, you can see a stain showing vegetative bacterial cells containing endospores. The endospores appear as distinct structures inside the cells because of their unique composition and staining properties. Because of these adaptations, endospores can sometimes survive for extremely long periods of time while remaining dormant. The remarkable resistance is one reason endospore forming bacteria are so important in microbiology, medicine, and sterilization. Endospores are not permanent dormant structures, although they can remain dormant for quite some time, as there's been honey found in the Egyptian pyramids where we were able to culture spores or regerminate spores that were in that honey. If environmental conditions improve, the endospore can return to active life through a process called germination. Favorable conditions, such as the presence of nutrients, water, and appropriate temperatures, trigger the endospore to begin reactivating. During germination, the protective structures surrounding the spore begin to break down. Water enters the core, and metabolic activity resumes. The cell then transitions back into a normal vegetative bacterial cell capable of growth, metabolism, and reproduction. So while sporulation allows bacteria to survive harsh conditions, germination allows them to rapidly take advantage of favorable conditions when the environment improves again. In many ways, the endospore acts like a survival state that allows the bacterium to pause life until conditions become suitable once more. Endospores are extremely important in both microbiology and medicine because their resistance makes them very difficult to destroy. In sterilization procedures, normal disinfectants or brief heating may kill active bacterial cells but fail to eliminate endospores. Because of this, special sterilization methods such as autoclaving are often required to reliably destroy endospore forming bacteria. Some medically important pathogens also produce endospores. Species of Bacillus and Clostridium can form highly resistant spores that survive in soils, surfaces, or medical environments until conditions allow them to germinate. Endospores also play major ecological roles in nature. They allow bacteria to persist through drought, nutrient limitation, heat stress, and other harsh environmental conditions that would kill many other microorganisms. This survival strategy helps certain bacteria remain in ecosystems for long periods of time, waiting for favorable conditions to return. So, although endospores are dormant structures, they have enormous impacts on disease, food safety, sterilization, and microbial survival across natural environments. All right. Thank you so much for attending this lecture, and I'll see you next time.